Mark Twain famously stated that there are three kinds of lies: lies, damned lies, and statistics. Now, I doubt that Mark Twain thought that all statistics are misleading, or that all statistics are evil or anything like that. But he was probably frustrated or concerned that statistics are sometimes used to mislead people, and he had a very creative way of saying it. He actually didn't make up this quote. He was quoting a previous source. Well, his quote inspired the title of this video's Lies, Damned Lies, and Origin of Life news articles. I don't believe that all Origin of Life news articles are misleading or evil, but I do believe that some of them are very misleading, and I'm going to share examples from this past week. So the University of Tokyo released a press release. The title was New Insight into the Possible Origins of Life. Experiment sheds light on the molecular evolution of RNA. This press release was itself based on a report of an experiment. The report was made in a scientific journal, and there the title was Evolutionary Transition from a Single RNA Replicator to a Multiple Replicator Network. Now, the press release did its work, and this news story, the science news story, was picked up and echoed in a number of scientific journals. Science Daily had this headline, New Insight into the Possible Origin of Life. All of these are based on this news report of this experiment. Science Alert, scientists create RNA that evolves on its own. That's an amazing claim. This could be how life on Earth started, they say. Health Thoroughfare, scientists create the first RNA molecule that replicates. If that was true, those scientists would deserve and almost certainly would get a Nobel Prize. We'll look and see whether or not that's true. Express stated origins of life breakthrough as missing puzzle piece of evolution found. Truly exciting and interesting. Engineering had this headline, a new RNA experiment could finally reveal the origin of life. Those are really big claims. So in this video, I want to do 5 things. I want to talk about why people care about this type of news, and then the key role of self-replicators for chemical evolution. That parts of this are a bit technical. They have to be in order to explain the issue. Number 3, what the articles claim or strongly imply. Number 4, what the experiment actually did, which most people would never pick up on from the news reports. And here some of these wild claims will be debunked and some concluding thoughts. So let's begin with why people care about this type of news. The people who wrote the news reports knew very well why people care about this type of news. The original press release said, life has many big questions, not least being, where did we come from? So they're claiming that this scientific experiment that was reported on might help people figure out one of the most fundamental questions of life, a big part of any worldview. Where did we come from? Interesting engineering in their news report said humankind has always been looking for an answer to the question of how life began and coming up with different theories on the origin of life. That's true. And then a quote from one of the people involved in the research, Professor Mizuki, said, the results could be a clue to solving the ultimate question that human beings have been asking for thousands of years. What are the origins of life? And lots of people do think about this. Even children begin to ask, where did I come from? Where did we come from? Where did mommy and daddy come from? Were we created by God or by unguided natural forces and processes? Those are normally the two big options. I'm not saying they're theoretically the only options, but those are the main ones that people are normally considering and thinking about, and what a huge difference it makes if we believe one or the other. If we believe that we were created by God, then that implies that there was a purpose for our existence, probably some kind of a plan. It opens the door to the possibility that this God might still want to interact with us. It makes it seem possible that this God could also have plans for what happens to us after we die. But if unguided natural forces and processes are the whole story for where we came from, then all of that seems less likely. It seems like there's no ultimate purpose, no purpose from beyond ourselves for why we're here, and no reason to think that there'll be anything left after we die. So I realize that the origin of life issue, and this is, and especially the scientific experiment by itself, doesn't directly answer this huge question, but it's related to this huge question, and that's why people care about this type of news. That's why I care about it. And yes, some people also are just into the science. They like the technical stuff. I also like the science. I've loved science my whole life, but this is why this type of news is of interest to more than a tiny group in a very specialized technical field. So the alternative to God creating life is life being created by solely natural forces. The theoretical process, if life is created by solely natural forces, the theoretical process involved is often called either chemical evolution or abiogenesis, but sometimes it's just lumped in with a broad view of evolution. That's what happens in a lot of these articles. That's fine. I think people know what they're talking about when they say evolution, but more specifically, it would be abiogenesis or chemical evolution. So next, let's talk about the key role of self-replicators for chemical evolution. This part is a little bit more technical. By God's grace, I think I have a gift of explaining complex things to people, so I hope this will be helpful. If you already know all this information, then please bear with me. I'm sorry that it has to be repeated. And here I'm going to let you see me for just a few minutes. Hold on here. There I am. Okay. So chemical evolution, one definition we could give is that it is an unguided process based on the laws of physics and chemistry and random events, with natural selection working on self-replicating pre-cell chemical systems. That's a mouthful. Let's see if we can think about it a little bit more and what it means. So self-replicating pre-cell chemical systems. In other words, before there was the first living cell, in order for there to be chemical evolution, there had to be either individual chemicals or groups of chemicals that could make copies of themselves. So this is what we are trying to explain. How to get from the types of chemicals that could be found on prebiotic Earth, in other words, these chemicals wouldn't come from any living thing, any living cell, to the first living cell. So that's the goal, is to explain this. That's the goal of chemical evolution or abiogenesis. And this process involves a supermassive increase in functional complexity. That's because the simplest cell is not simple. There have been experiments and also theoretical studies on the simplest possible living cell, and it contains a massive amount of functional information and a large number of amazing molecular machines inside. It is not simple at all. It is mind-blowingly complex, but the chemicals are relatively huge amount more simple. So how do you get this increase in functional complexity? And okay, let's see. Here we go. So chemical evolution requires that thousands of relatively small steps happen in between the initial chemicals in the first living cell. The theory of evolution requires that any increase in complexity be a relatively small increase. So no evolutionary scientist thinks that, for instance, the human eye just popped into existence in one step. They think that there were a huge number of steps, starting with maybe photosensitive light spots. Even before that, somehow there had to be photoreceptors inside of cells. And so also in between these simple chemicals in a cell, there would have needed to been, really considering the complexity of the simplest possible cell, there would have needed to been many thousands of steps. And for this to work, at the very least, each step must consist of a self-replicating molecule or system of molecules. If it can't reproduce itself, then it's a dead end when it comes to evolution. It might be some interesting chemistry, but it would be useless for evolution. It also has to be, each step has to be increasingly complex on average. So for instance, maybe step 2000 to step 2001 was not increasingly complex, but on average, it has to get more and more complex and be increasingly cell-like on average, or you would never get to a living cell. Now, here's the big problem that we're going to focus on. It's not the only problem, but it's a really big one, is that all of these steps have to involve self-replicating molecule or system of molecules. They have to be able to reproduce, or they are just totally irrelevant to chemical evolution, to the theory of life coming from natural processes that are unguided. Now, what do we mean by self-replicating? This is important. You might want to read this a couple of times. By self-replicating, we mean a molecule or system of molecules that completely replicates itself using material that would have been available. So if the system only replicates half of itself, but you need the whole system to continue the process of self-replication, that would be useless. It has to completely replicate itself, or more technically, it has to replicate enough of itself to continue being able to do self-replication. And it has to use material that would have been available. In other words, if you have a cell and all it needs is one more molecule to become a self-replicator, that's not realistic because you wouldn't have had cells on the ancient Earth. It has to use material that it would have had. Now, that's going to be debatable. What type of chemicals would have been available before there was life? And would there have been amino acids? Would there have been things like the nucleotide bases? Would there have been sugars? And would these chemicals have existed in sufficient concentrations, in sufficient purities, with the right chirality, that they would have had to have to be useful and relevant for an evolutionary process? Dr. James Tour has an excellent series of video addressing why these chemicals probably would not have been available. But let's assume that at least some of them were, just for the sake of argument. So many thousands of such increasingly complex self-replicators would have been needed. How many have been found so far in nature and or created in a lab? Well, I made a video, and I'll include a link for it in the description to this one, where I claim that the answer is 0, and I defend that claim. But if this new news article, if this group of news articles is correct, then it's gone from 0 to 1, if we believe the recent science articles. And while that's less than the thousands that are needed, it would still be a huge breakthrough. The specific self-replicator that they claim to have made wouldn't be all the way at the top near the cell. Neither would it be all the way near the bottom. It might have fell somewhere in the middle of the thousands that are needed in terms of increasing complexity and increasing cell likeness. But to find even one would be a huge scientific achievement. Now let's talk about what the articles claim or strongly imply. So This is the original news release. They say that the researchers, for the first time, see, I'm saying 0 to 1. That's not me saying this is number 1. This is them saying it's number 1. For the first time, it's been able to create an RNA molecule that replicates, diversifies, and develops complexity following Darwinian evolution. Excuse me. This is minimally a misleading, gross exaggeration. Most people will probably read this when it says an RNA molecule that replicates. They will probably read this to mean self-replicates, since it sounds like the RNA molecule is what does the job of replicating, and also because it would not be newsworthy if the RNA molecule was replicated with the normal cellular machinery found in all living cells. So this is implying that they have made, for the first time, one of these self-replicating molecules, and it doesn't necessarily have to be one that was actually part of the evolutionary process, but it would be similar. It would be an example of the type of molecule that might have existed in the evolutionary process. And the articles very clearly are making this type of claim and this type of assumption. Notice that the first paragraph in the news release says this has provided the first empirical evidence that simple biological molecules can lead to the emergence of complex lifelike systems. Wow, stop and think about this. Prior to this experiment, there has been zero empirical evidence for chemical evolution. That's what's implied by the articles of these science reports. In these articles, in these science reports, by these authors who seem to believe, they seem to be positive about the ideal of the theory of evolution. And they're saying, basically, up to now, we haven't had any empirical evidence for this chemical evolution. I basically agree that there hasn't been any empirical evidence up to now that would actually meaningfully support the ideal of chemical evolution. But I think that they're wrong to claim that this new experiment is the first. I don't think this experiment should count as evidence for chemical evolution. Science Daily uncritically copied this paragraph into their own short article. Did they stop to question it? If they did, you can't tell. And then Science Alert, they say that this RNA can replicate all on its own. What an amazing claim. And they imply that it can carry out this replication task without a supporting cast of highly specialized organic materials such as DNA or proteins to assist. We're going to see that this is just straight out false when we look at the details of the experiment. Science Tech Daily said that this is the first. This is a article in Health Thoroughfare, but they are quoting Science Tech Daily, and they say with approval that this experiment produced the first RNA molecule that can replicate itself. Replicate itself. That sure sounds like a claim of self-replicating RNA to me. We'll see soon whether or not that claim is justified. And then an article in the Express said an RNA molecule that can self-replicate, talking about this experiment. So these are huge claims. If it's true, these scientists would be worthy of Nobel Prizes. We'll see soon whether or not it's true. Let's talk next about what the experiment actually did, and we'll debunk some of these wild claims. Now, before we talk about the experiment, we have to talk about, at least I think it will be helpful to talk about how RNA is normally made in cells and how virus RNA is normally copied. So in cells, RNA is not made from other RNA. RNA is not copied. Rather, RNA is made from DNA. So you start with DNA, and then you have a thing called a process called transcription, and it uses an RNA polymerase. This is a complex molecular machine with several protein subunits, and this complex molecular machine makes a strand of RNA from a portion of DNA. Now, where did the RNA polymerase itself come from, this complex molecular machine? Well, the instructions, the information to build an RNA polymerase is in the RNA. It's in some of the RNA in the cell, and it uses a process called translation. Now, translation has a whole system of complex molecules, including ribosomes, and this process of translation takes the information from the RNA and builds protein and protein machines, molecular machines, from using that information. And one of the machines it builds is the RNA polymerase. So we have a little bit of a chicken and an egg situation going on here. But here's the key point. While RNA plays a limited role in producing itself, it does not self-produce. It uses a complex system of many parts in the cell in order to produce itself normally in cells. Now let's talk about how some RNA viruses hijacked a cell to copy their RNA. This is going to be very similar to what happens in the experiment. So, part one, you have RNA in a virus. Every virus contains genetic information, either in RNA or in DNA. So some viruses start with RNA in the virus, and this RNA contains code for perhaps a number of things, but one of the things it has code for is a different type of RNA polymerase called an RNA-dependent RNA polymerase. That's a mouthful. And what it is it's a molecular machine, a polymerase, that instead of using DNA to make RNA, it uses RNA to make RNA. So let's go back here. Then the virus, this RNA in the virus, uses the infected cell's translation system. So the cell doesn't know the difference between virus RNA and its own RNA. So it sees this RNA and it uses it to make this special molecular machine, this RNA polymerase. Now, how does the cell do that? Well, it uses the cell's translation system, which is a whole system of complex molecules, including ribosomes. We'll look at that in a minute. And then, using the cell's system of complex molecules, including ribosomes, the cell's translation system, it produces an RNA-dependent RNA polymerase, which is also called an RNA replicase. And then here's part two. So you have RNA in the virus still, but now you have an RNA-dependent RNA polymerase, and that uses the information in the RNA in the virus to make new copies of RNA from the virus, new copies of virus RNA. This is a simplified explanation, but it's accurate. It doesn't have all the details, but it's accurate. Now here's the key point. Viruses do reproduce, but they are not self-replicators. And I've read a lot of stuff about viruses and cells and self-replication. I don't think anyone calls them self-replicators. They are not self-replicators because they need the whole translation system of a living cell in order to reproduce. There's a vast number of different types of viruses. Viruses don't just infect people, they infect all types of living things. They infect plants, they infect all kinds of animals, they even infect bacteria. Different types of viruses infect different hosts. None of these types of viruses, no virus we have ever found, can self-replicate. None of them can produce a copy of themselves outside of a living cell. All of them have to invade a living cell in order to reproduce. So it's important to keep that in mind. Now, what happened in the experiment? And again, this is a simplified summary that, while simplified, I believe is accurate in terms of the issue of self-replication. You can go look up the original experiment yourself and read about it if you want. So they start with RNA that they got from an RNA virus. Why do they start with that? The answer is that this virus RNA includes the coded instructions to build a RNA-dependent RNA polymerase, also known as an RNA replicase, because they need this to make copies of the RNA. So that's what they start with. But the RNA virus is not the only important ingredient they put into their test tube. So we'll talk about the other ingredients in just a minute. And that's important. It's vital to understand what's going on. Now, what would have happened if they put this virus RNA into a test tube of pure water, or if they put it into a test tube with what they imagine to maybe be the ancient, some people have described it as the prebiotic soup, on the organic soup that people imagine might have been on the earth. I don't know if such a thing existed, but let's say that they put it in the test tube together with nucleotides that make up RNA and the right kind of sugar that also makes it. In other words, they put in the pieces of RNA that would have to be assembled to make an RNA molecule. People can debate how likely those pieces would have existed, again, in the right purity, in the right concentrations, with the right chirality. That's a huge issue with chirality instead of being racemic. But let's say that we give them that, and they put the RNA in the test tube with that stuff. What would have happened? Well, at first, nothing would happen, and then over a relatively short time, the RNA would break up and disintegrate. We know that RNA is not stable in water. It doesn't instantly break up, but it doesn't last very long. DNA is more stable. DNA is the long-term storage molecule for information. RNA doesn't have to stay around a long time to do its job. And unless it's bound up with other molecules, it doesn't usually stay around very long. And it would do nothing. It would be totally useless, a very boring experiment. But instead of putting in just the RNA and the chemical components needed for more RNA, they took a whole translation system from a bacterial cell and put that in tiny bubbles of water together with the virus RNA. So that's what they put into the test tube. So let's talk about this, what this is, a whole translation system. It includes, in this particular case, dozens of enzymes, 46 tRNAs. That's 46 different types of tRNA. It's not 46 of the same molecule. And ribosomes. Let's look at what these things are quickly. Enzymes are, all of them are complex. All of them involve a specific sequence of amino acids that were coded for in the DNA of the cell. And they do specific chemical functions. And these things are extraordinarily unlikely to have existed on a prebiotic earth. And then they also included these 46 different tRNA molecules. These are an essential part of the cell's translation system. You have to have a lot of different ones for it to work. And these are also very complex. These are also extraordinarily unlikely to have been found on the prebiotic earth. Super extraordinarily unlikely. And then they put in ribosomes. This is not a complete ribosome. This is a subunit, actually not the largest subunit, of a bacterial ribosome. The proteins are in blue. The orangish color is the RNA. It's very complex. When it's put together with its other pieces, it does an amazing job. I encourage you to look up on YouTube animated videos that show protein synthesis in cells, or you could look up translation in cells, or ribosomes, how they work in cells. There are some beautiful and fascinating videos that show this process and also show how amazingly complex it is. So this whole translation system they got out of a cell, all of these parts were made in living cells. They take them out of the cell and then they put it in the water together with the RNA from the virus, and more. They would also need ATP. ATP is an energy molecule. Some of these chemical machines, some of these microscopic little machines that are so amazing, some of them need energy to do their job. And this energy comes from a couple of different sources in the cell. One of the most common is the ATP molecule. The ATP molecule itself is produced in the cell by an amazing group of machines, one of which is ATP synthase. If you look up animated videos on ATP synthase, you can also see that. It's astounding. So they take this whole translation system and they put it in the test tube with their RNA that they got from the virus. Now they are trying to explain the origin of life, but all of these ingredients were produced in living cells. None of them were produced with prebiotic methods. To me, that seems like cheating, really big cheating. Like if Einstein was still alive and you could get him to take your high school physics test for you, that level of cheating, in my opinion. If they want to have an experiment that says something about the origin of life. Now, they could do this experiment for other purposes, and it would be fine. But to be relevant to the origin of life, wow. Okay, so back to the experiment. So they take the virus RNA, the RNA that they got from the virus. I think they modified it a little bit. My guess is that they mostly cut out stuff they didn't need. I'm not sure about those details. But they put that together with a whole translation system for bacteria. All those different parts that I just showed you. And they put it in a test tube. The test tube has water and oil. The water makes little tiny drops in the oil. And that way, all of these chemicals can stay close together. By the way, oil would also not have been available on the prebiotic earth because oil comes from living things. But that's a very minor point. And then using information from the virus RNA, the translation system that we just saw made RNA replicase, which then made copies of the original virus RNA. But the translation system was not copied. So, this is not self-replication because, one, only the virus RNA and the replicases are replicated. The translation system is not replicated. And they did this experiment over and over again. They did generations of this experiment. And every time they put it in a new test tube, they had to add new translation systems because the translation system is not being copied in this experiment. And then number two, the RNA replicase system cannot replicate itself without the translation systems that were made by living cells. So to claim that this is an example of RNA self-replicating is just, to me, it's just outrageously misleading and just false. Just false. Claiming that the scientists created a self-replicating RNA or a self-replicating molecular system would be like taking instructions on paper to build a photocopier and then copying them with the photocopier and then claiming that you had magical self-copying photocopier plans. But the plans did not build the photocopier, and the factory that built the photocopier was never copied. Let me explain this analogy. You probably got it, but just to make sure. The instructions on paper to build the photocopier, that's like the virus RNA that contained instructions to build RNA replicase, okay? And then the photocopier is like the RNA replicase. It can copy the plans, the paper plans, but it cannot make copies of itself, much less of the translation system that made it. And then the factory in this analogy represents the whole translation system that was made in living cells. And a factory is a very good analogy. I'm not the first person to think of it because there are many molecular machines working together to do something. And none of that is copied in this experiment. They just take it over and over and over again from cells that were living when they made this material, the translation system. So here's the analogy again. Claiming that the scientists created self-replicating RNA or a self-replicating molecular system would be like taking instructions to build a photocopier and copying them with the photocopier, and then claiming that you had magical self-copying photocopier plans. But the plans did not build the photocopier, and the factory that built the photocopier was never copied. So you do not have a self-replicating system. The plans didn't copy themselves. They were copied with the photocopier. The photocopier didn't make itself. It was made in a photocopy factory, and that's similar to what's going on. Now let's quickly go back and look at some of those wildly misleading claims from a few of the Science News articles. Not all of them, just a few quickly. This one speaks about RNA that can replicate all on its own, and it strongly implies that it does this task without a supporting cast of highly specialized organic materials. Baloney! That is totally false. I hope you see why that's totally false now that I've explained to you what happened in the experiment. Now I encourage you to read the original papers. The first one isn't that hard to find, but that references another one, which references another one. It's a little bit hard to tease it all out, but this is just outright misleading. And then PixxlTeufel spoke about an RNA molecule that can replicate itself. No, they did not make an RNA molecule that could replicate itself. That's wildly misleading. I hope you can see that now. And then the Express said an RNA molecule that can self-replicate. Totally false. I mean, I'm sure these people, I don't want to call it a lie because I seriously doubt the intent. Normally, when we say somebody's lying, we mean not only that they're giving false information, but that they know that they're giving false information. But these people are writing Science News articles. They should be careful. They should research this. They should ask questions. They should have been able to figure this out. And this is just false. It's just a false statement. It's very misleading. Now, some concluding thoughts. The articles also claim that in the experiment, the RNA evolved. That's a big part of the claim. But without self-replication, there can be no evolution in the biologically relevant meaning of evolution. If you don't have self-replication, you definitely do not have evolution. But, number two, there are other problems with this claim, with the claim that there is evolution. And these problems are examined in two excellent articles at Evolution News. Both of them are by Brian Miller. The first article is what alerted me to these news reports and prompted me to make this video. These are both good articles. Unlike the ones I talked about earlier in the evidence, these carefully discuss what actually happened in the experiment. They're excellent. They're helpful. They're both found at evolutionnews.org. I strongly recommend that organization and that site as a good place to get organization on the origin of life and other issues. Now, why does this bother me? You know, it might seem like a really esoteric issue, but it's not. This type of misleading material robs God of worship and robs some people of faith in God. When we see something amazing, like creatures who are able to reproduce, the most awe-inspiring is when we see babies. And in the last couple of weeks, two young couples I know have just had babies. One of them is are members of our church family. Another one is a member of my extended family. My niece and her husband just had their first child. It's awe-inspiring, and it should motivate us to worship God, who made creatures that are amazing. And one of the amazing things about us is that we can reproduce other creatures that are like us. But if people believe that evolution is responsible for all of this, then they are less likely to worship God when they see flowers making flowers and butterflies making butterflies. But we should worship God when we see that happening. And then quite a few people who reject Christianity list science as one of the reasons. And this is the type of thing they mean. They read this type of material, they soak it in uncritically. They think that science has explained where we came from without needing God in the picture, and it undermines their faith, and that is tragic. I want to close with a short prayer. Heavenly Father, I thank you that you made all of the amazing types of life on this planet, that you designed them. Lord, it shows how intelligent you are, how wise you are, how great you are. Thank you, Lord. Lord, I pray that people will see the truth that we have a great Creator and will come to know you and will worship you. In Jesus' name, amen.